Good afternoon, everybody. I, Dr. Vivek Srivastav, Associate Professor, School of Pharmacy, welcomes all of you who have joined today for the webinar. Honorable Dr. M.N. Patel, sir, Provost, Parul University. Respected Dr. Abhay Dharamsi, sir, Dean, Faculty of Pharmacy and Principal, Parul Institute of Pharmacy. Respected Dr. Vandana Patel, Madam, Professor and Dean, Doctoral Studies. Dr. G. Chakravarti, Principal, Parul Institute of Pharmacy and Research. Dr. Lita Jha, Madam, Principal, School of Pharmacy. And please kindly join me in welcoming Dr. K.S. Jagannathan, sir, Deputy Director, Serum Institute of India. Dr. K.S. Jagannathan, sir, is working as Deputy Director and heads the production department since November 2017 at Serum Institute of India, and he is focusing on formulation development, commercial manufacturing, and troubleshooting activities for vaccine and vaccine adjuvants. He obtained his PhD in pharmaceutical sciences under the guidance of Professor S.P. Vyas from Dr. Hari Singh Gaur University, Sagar, in collaboration with National Institute of Immunology, New Delhi. He did his postdoctoral studies at Tokyo University, Japan, and worked in collaboration with Kowa Pharmaceutical Limited, Japan. Prior to joining Serum, he worked for Sanofi for about 14 years on various projects related to biologics, vaccines, monoclonal antibodies, and protein and drug delivery. Jagannathan, sir, is a recipient of Best Scientist Award by Ministry of Science and Technology, Government of India, and was chosen as a key scientist to attend the Nobel Laureate meeting at Germany. He is also a recipient of several key awards like BioAsia Innovation Award by Nobel Laureate, Professor Martin Evans, UGC JRF, AICTE NDF Young Scientist Award by DST, Government of India, JST Postdoctoral Fellowship Award by Government of Japan, and Best Employee Award that is in Platinum category by Sanofi Group of Companies. He has several referred publications in international journals, besides oral presentation at international conferences and symposia as a speaker. To his credit, including many US patents, he has more than 20 years of experience in pharma industry at various roles of product development and operations. And in fact, so far as vaccine are concerned, so vaccines is a word which is on the tip of everybody nowadays because of the corona, and everybody is looking at the vaccines very hopefully. So if we see the newspaper or we go to the television news, so everywhere everybody is discussing. And today we have with us Dr. K.S. Jagannathan, sir. So it's a good opportunity to hear from him about the multivalent vaccines. So sir, please, I request you to give your deliberations on the multivalent vaccines, sir.
Thank you, Vivek. Thank you, everyone. It's my honor to present today. And the topic for my today's presentation is developing formulations for game-changing multivalent vaccines and its cost-effective strategies. So I'm not going to give something confidential which are which are integrated in our manufacturing process. I have just made this presentation with some case studies as a and also it's a generic presentation. So if you have any specific questions, feel free to ask me at the end of my presentations. So before I go in detail about my the different formulation strategies, so we should have like short introduction about vaccines. So first, what are vaccines? As Dr. Vivek said, vaccines nowadays we are getting more visibility into vaccines. So what are vaccines? So vaccines, we know obviously it saves life, preserve good health, maintain a high quality life. So vaccination stands one of the most successful public health measures in the last century. So vaccines have provided some of the greatest success in the history of medicine. You know very well eradication of smallpox and eradication of polio. And it has to be most cost effective, safe and powerful tool of medicine. And it prevents suffering, disability and death from infectious disease. So I just wanted to start my presentation with some pre-vaccines events. So still there are some guys, so they are little hesitant to take vaccines So first they should understand what vaccine does, and we should start with some case study. So let me present with some previous events which has happened in our life. So if you go for vaccines, what happens? Birth defects caused by rubella. Okay, so the children crippled by polio, horrific sound of babies struggling with whooping cough, neonatal mortality by tetanus, and terrible skins of hydrophobia. So now every year up to 5 million children's life are saved by immunization. So these things, I have some couple of slides in my next, presentation. So I will just present you like how vaccine has helped in avoiding these diseases. So vaccine is certainly important, and we should take, and it has already saved people. And if we have to be, we have to be cautious in, making comments against vaccines. So I will just give some case studies, then you conclude how vaccine helps. So coming to this slide, this is the disease case in US at 20th century, before advent of vaccines and after utilization of vaccines. You see the column that is different disease: smallpox, poliomyelitis, diphtheria, Haemophilus influenza, measles, mumps, pertussis, rubella, and tetanus. So the estimated pre-vaccine cases in 20th century. So you have all these diseases in million. So the deaths in 2017. So you see smallpox and poliomyelitis 0, and diphtheria only 2 cases, and Haemophilus influenza is 22, and measles 36, mumps 236, and pertussis 6,632. Rubella and tetanus is very negligible. You just compare the deaths in 2017 and the earlier, in 20th century. So you just compare. So how this has reduced? So you have an answer. It's due to vaccines. So these vaccines are already established in market, and many companies started manufacturing. So now we are at this stage due to vaccines. So this is the case study just to compare how vaccination helps in public. So I will just give India's polio fact sheet in India. So India also, though it's a polio-free country, there are some cases in outside the countries, like limited. So first let us see the polio fact sheet in India. So in 1985 we had 1,50,000 cases, and followed by started decreasing. And the last case what we got, the last case of polio what we got in India was in 2011. So that is on 13th January. So we had the type 1 was noticed in 2011, and type 2 was noticed in 99, and type 3 was in 2010. So last case was 13 January 2011. So this is the major milestone in the history of polio eradication in India, and we expect to have polio-free world by 2023. So this can be achieved only by vaccines. Okay, so if you don't have vaccines, we are not going to be in this case. So vaccine obviously helps in preventing death, and vaccination is very important, especially at the cases like we have this, like COVID. So how vaccination helps? This is just case studies. Okay, so though this vaccine, despite our success, we realize that the battle against disease will probably never end. And since new challenges continuously appear, example, anthrax, bird flu, TSC, BSC, chikungunya, dengue, and SARS, now COVID-19. So our struggle is going to continue, and we have to upgrade, and we need to have vaccines to avoid and to sustain, to survive in market. So now let us understand, before we go in about formulation strategies, I just wanted to present the vaccine scenario past and present. So if you go back 30 years back, so when Louis Pasteur started working, and on the vaccine scenario at earlier stage, so the vaccine manufacturing facility, there was no defined GMP facility, and there was no written regulatory guidelines, and they used to inject directly live virus inoculation, and no complicated process, and everyone used to think it's a God's punishment. So if you have some infections, so they used to call it God's punishment. So no one is aware about the science, what is virus, fungal, and this type of things were not aware. So everyone used to believe it's God's punishment, and there was no scientific work as we have at this stage. and causes of disease was not known. The root of, the root cause of the infection was not known earlier, and obviously there was no vaccines for many infections. So if you come back to the vaccine scenario, the present one, so we have defined facility, GMP defined facility, so where we will be able to manufacture. This is the right hand side. If you see the earlier Louis Pasteur period, how lab used to be, and if you see our industries now, we have completely automatic, completely closed operations. So we have defined the GMP facility. And if you, I will just compare the GMP facilities what we had 10 years back. When I started career, we used to have the normal filling line, which is we felt actually which is hi-fi and the best one. So way forward, we started having O wraps. Then later we started having C wraps. Now we have isolator. So we get, we have to migrate into the new systems. So the 10 years back, the systems what we had, when I started my career 20 years back, the system what we had and the system what we have is completely different. So we have to get upgraded with the current systems, and we have to upgrade our facilities. The facility what we were using 10 years back cannot be used. So we have to develop new facilities which has to fulfill the current regulatory requirements. So we have to, it's like evolving process. So we have defined the GMP facilities from each regulatory body. Say for example, US FDA, India, for example, CDSCO, or in European guidelines or Japanese. Okay, so now there is no vaccine where we inject live virus. So there is no live virus inoculation as they used to do earlier. And we have NRS testing laboratory. For example, in India, we manufacture our vaccines. And once you manufacture, we will test at our end, ensure that the vaccine that we manufactured is will fulfill the requirements, the defined quality requirements, and also the complies with the pharmacopoeia which we integrate in our testing system. So once this testing process is completed, we send our vaccines to NRA. Say for example, in India we had a CRA Kasoli. So we send our vaccines to CRA Kasoli. They test again, they give green signal to go ahead and release our product to market. So this is how we follow. We have defined NRA system to test our vaccines before we send outside our facility. So we test each vaccines. It is not only COVID vaccines. So any vaccine that goes out from our facility, it has to be released by NRA, CRA Kasoli in India. So we do have defined clinical path for any vaccines. So vaccines, you have two paths. One is the completely new vaccines where you have to follow the classical regulatory path, where you have to go for safety studies, few studies in animal model. It's called a toxicological study. So once you are okay, and if you prove that the toxicology studies are fine, it is not giving any toxic effect, then we go with this data and present to DCGI, and they give green signal to start clinical phase 1 study. That is phase 1 study is nothing but safety studies with minimum subjects. Once we are okay, and if we are successful in clinical phase 1, we go for phase 2 study. It is partly safety studies, and also we perform the dose response in the clinical phase 2 studies. If everything is fine in clinical phase 2, then we go for clinical phase 3 study with more subjects in thousands. So once we are done with clinical phase 3, then we launch our product, and still we continue our clinical trials to monitor any adverse events. So that is called clinical phase 4. Now we have defined guidelines on clinical trials as well, which we never used to have earlier. And the vaccine always 6 to 95% coverage, and which are well defined actually how to calculate and how it is being done. And now we are in a situation. If we ask me questions, microorganism or man, who is going to be the winner? Now we are in a stage to say, man. So it's a proud moment. Earlier, microorganisms used to be the winner. So now we have good science, and we have good capabilities. We have good scientists. So we still we are confident that we can develop vaccine and we can face any organisms. So still we can compete with microorganisms, and still we can win. So now, if you go for this vaccines disease, it keeps increasing. If you go for like 15 years back, so we used to have tetanus toxoid, then diphtheria toxoid, then pertussis, Then WHO started recommending, recommend to develop combo vaccine. It is nothing but combination vaccine. So where if you are having 5 infectious disease, so instead of taking 5 injections and booster 3 example, then 5 into 3, how many boosters? So now they started recommending to develop combo vaccine, where we develop combination vaccines. Now there are vaccines with 5 antigen in one vaccine formulation, and 7 or 8 vaccines also is being developed currently. So now every company, they concentrate in developing combo vaccines, and we have up to pentavalent vaccine in with WHO pre-qualification, and we are also developing hexavalent and heptavalent vaccine in our facility. So this is how it revolves actually. So now we moved from monovalent to 6 antigen in one vaccine. So developing combo vaccine is another area which was never explored in earlier scenario. Then cold chain management depends on vaccine. Some vaccines it has to be kept at -80 and -20, and most of the vaccines are between 2 and 8 degrees Celsius. So this cold chain management is well defined and and it is we have defined systems in our current practice. So you see the difference, where do we stand 30 years back and where are we? So it shows that the vaccine is revolving, and it is absolutely we can see the difference actually, the earlier scenario and current scenario. Okay, so now let me start with some the final product action vaccines. You have 2 parts. One is the drug substance and the drug product. Drug substance is nothing but active pharmaceutical ingredient which is in pharma, it's common word API. So in vaccine industry, we have to call it as drug substance, that is nothing but bulk. So the active pharmaceutical ingredient API. So once this API is in place, then we have to convert to formulations. So everyone thinks that if it is combo vaccine, you just mix components of A, B, C, then you get C. So it's a final formulation. So it is not so. So it's again formulation science. Some vaccines are with aluminum adjuvants. So we have conventional adjuvants. So there are only adjuvant approved by US FDA or WHO is aluminum adjuvants. There are many adjuvants are being developed currently to improve the immune system's immunity. And the conventional adjuvants are aluminum adjuvants. Then we do have some lyophilized products, like for example MMR. If the vaccine is not soluble in liquid form, then we have options to develop lyophilized. So what is lyophilized? We just remove the moisture inside, or we remove the water. So I'm sure you would have studied about lyophilization process, which I'm not going to discuss in detail. So we have NDDS, novel drug delivery systems. So here you have many scientists are trying to develop the new adjuvants, so that it can follow both MSC1 and MSC2 complex to give coverage, the broad immunity. That is nothing but mucosal immunity plus humoral immunity, that is IgG. So in addition, cell-mediated immunity. So such NDDS-based systems are either microparticles or monoparticles, or sometimes dendrimer, soluble microneedle coated with some nanoparticle. What happens? You have soluble microneedle, then you coat with nanoparticles. Then you just press as transdermal patch, then the nanoparticles delivers the antigen. So this is how it happens. Now it evolves. Then there are vesicular systems like liposomes or neosomes. So there are different systems under development. So here I will just present 2 systems which was integrated in my earlier work. So with this, I will just give some the case studies with industry, linking industry practice. So what are the ideal requirements of vaccine formulations? So which is very important when you develop formulations. The first and foremost, the key requirement in vaccine formulation is stability. So the stability, your vaccine can be either liquid or lyophilized. So as a formulation scientist, we should have our preferred option to have liquid vaccines, because if it is lyophilized, you need to buy lyophilizer, and which will add the cost to your product. That is nothing but COGM. So again, by the time you bring it to market, so your liquid, the formulation cost plus lyophilization cost has to be integrated. So when you come to market, it is going to be very expensive. So our preferred option to have any vaccine in liquid form, in case of any challenges with respect to stability, then you are left with no other options. You have to go for lyophilized vaccine. So you have to keep remembering, you should remember when you go for lyophilized formulations, the COGM increases. Then the second part is very important, selection of excipient. So you have, options to choose excipients. You have 1,000 different excipients. How smart, how smart you are, how to choose these excipients, you need to consider multi-factors. Say, for example, if you want your vaccines to be sold in Japan, there are some excipients which are not acceptable in Japan. Say, for example, beta-cyclodextrin. And the same beta-cyclodextrin is acceptable and approved in EU or in US market. So if your product is intended to be used in Japan, you should not choose such excipients during your development. Once you develop, once you complete your clinical study, again, you cannot change your excipient, you cannot change your product composition, you cannot change your process. So then in case if you want to change, then you have to go back to your zero. Like first step, you have to start fresh clinical trial and ensure the safety and efficacy, then you come to market. So you have to be very careful and smart enough to take right decision, right excipients to ensure maximum stability. So there are some excipients which are acceptable in US and US market, which are not acceptable in EU market. So if you have to target both the market, US and EU, so you choose the excipient which are acceptable in all markets. Okay, then easy operations. So when you develop your product, you have developmental scientists with post graduation and PhD. So they are very good in defining process and they care. And the duration, times, all these things are not bothered when you develop. So when you go for actual commercial operations, so it's not like R&D. So you need different scale. And the guy who operates, he will not be scientist. He is not going to be PhD. So it may be it may be manufactured by normal technician. So you define your process. It has to be very simple. And if you have a process, for example, in say one product, there are some companies, they have lyophilization cycle up to 3 days. And there are some companies they complete in 1 day. So if you consider the lean principle scientifically, and also there are multi-factors, to integrate your process step. So if you consider the lean principle and process step, it has to be easy operations, and it has to be short test run. If you have for 3 days, your lyophilizer cost, your manpower, your facility, everything will be wasted. Your production capacity will come down. So you have to develop a product, and it has to be short. We should not have any unnecessary steps. In lean principle, it is called Muda. So we should not have such things in our development and the scale-up process. So scale-up is again really challenge. Sometimes what happens when we make a batch at 1 liter or 10 liter, so it works fantastic. It gives very good results with respect to stability, safety. And when you go for clinical phase 3, say for example, we manufacture at 400 liter. Then when you go for commercial launch and the continuous commercial batches, it has to be at, like we manufacture up to 3000 liter. So in this scenario, how do you ensure the batch that you manufacture for toxicological studies, the batch that you manufacture for clinical phase 1, phase 2, phase 3 and commercial and way forward further scale up? So you have to introduce something additional characterization which are not defined by any regulatory bodies. This comes with your experience. Your development scientist and your team has to define what are the identify the additional character, additional tests which can there are defined tests that has to be tested and released. So in addition to the defined pharmacopoeial test, the additional characterization test has to be introduced to ensure that the product you manufacture at R&D scale and commercial scale are comparable. So this all, like, there is no written publication, there is no written guidelines. So each manufacturers have to define their characterization, which will be very useful to strengthen your CMC data. CMC is nothing but chemistry, manufacturing and control and industry operations. So unless you have this strong CMC package, it is not going, it's not going to be smooth way to define your product when you go for commercial scale. If they are not satisfied, then they will not approve your, higher size and they may ask for additional clinical trial. So we cannot afford to go for further clinical trial unless you know about your process, unless you characterize your product. So this is very important, and if you miss and if you go for another clinical trial, it's going to cost your company $1 million and anyway further delay. So to avoid, you need to have smart team to identify the right tests that are very important to show that product you manufactured in R&D scale, clinical and also the commercial are comparable and safe, and also stability is established, including efficacy. So the scale up is going to be challenges. And sometimes what happens once you have you have scaled up to 3,000 liter, and you may extend to another facility. Again, the tech transfer from facility 1 to facility 2 is not going to be smooth, even if you have apple to apple equipments. So here your knowledge is very important. So you need to have complete process knowledge to understand the product behavior to so that you will be able to fix the issues on same day. Assume if you do R&D and if you kill time for 3 months, we cannot keep our commercial facility for ideal for 3 months. Then again they will fire you. So we have to mind it. This this all your process knowledge and the how smart you define and you manufacture, everything is linked with COGM and company success. And if you have to be competent enough to be with other manufacturers or other competitors, so we need to have smart facility and smart scientists who can fix the issue on time and ensure the product is manufactured with continuous consistency or robust process. And coming to the analytical methods availability, which is also very important. So you need to have right analytical tools to identify and test your methods, so which are very important. You need to have strong analytical team to study that the product that you manufacture or develop is the right product. So assume sometimes we have 3 processes, so if you have to choose one, so unless you have right analytical methods, you will not be able to take any decision. So the right analytical methods availability is very important. And when you go for clinical phase, you ensure that your test methods are validated before you start your clinical phase 3 study. So which is very important. And the next point is shelf-life, which is very important when you develop a vaccines. So you should target to have maximum shelf-life. Ideally, the vaccines are having about 2 to 5 years shelf-life. So at least you should have at least 24 months. Assume you make a product, if it is stable for only 6 months, then how it is going to be competitive? It's not easy process. If it is 6 months, by the time you test at your end, including in vivo studies, it's 2 months. Again, you send to CRA, Kesoli, they test, they take 2 months time. Then you with 2 months shelf life, if you go to market, I don't think it's feasible. So we need to have long shelf life, means you have to develop stable formulations, and through you have to perform stability studies to show that your product is stable up to X years. So which is very important. Again, the regulatory requirements. So again, this is country specific. So as I said, there are some excipients which are acceptable in X country and some are not acceptable in Y countries. So depending on the regulatory requirement, if you target to sell your product in X, Y, Z countries, so you have to align with their respective countries' requirements. So for example, we are supplying our product to more than 170 countries. So we fulfill the requirements from each country. So this is how we have to integrate during your development and commercial, which is country-specific requirement. Then obviously other product market demand is very important. You develop a product. So assume you start with the 10 projects, and when you go for clinical phase 3, it's going to be very expensive study. You cannot explore all 10 products in clinical phase 3. So you have to narrow to 1 or 2 products. So in this scenario, this market demand is very important to take decision. Assume you develop a product and you invest money up to say for example, if you are investing $100 million on facility. So later, if the product demand is only 5 million dose per month or 10 million dose per year, then you are going to keep this facility ideal for 9 months. So is it worth? So you have to consider the market demand when you develop your product. And when you buy your equipment, you have high speed machine, low speed machine. If your product demand is only 10 million, then you choose the filling line which are required for only 10 million dose. Don't buy the equipment which are required for 100 million per month. If you don't do, then it's going to be, if you don't integrate this in your product, then it's going to be setback for a company. So it's not ideal path. So you have to consider market demand and competitors who are developing, and IPR issues. So when you develop, if you integrate some process, ensure the other companies, you are not copying any other company's process. So if you are copying, then it's going to be challenged when you go for market, when you go in commercial manufacturing stage. So if you develop something, you go for patent, you patent your process, then you go to market. And as I said, if you have some facility, if you don't use for a whole year, and it is not good actually. So there is something in industry called OEE, operational efficiency. So if you are a good operational leader, so if you are running some facility, if you are responsible for 5 facilities or 10 facilities, so you need to review your scientist capability and facilities, how it is being utilized. So if I have to review, unless our facilities are like 80% occupied, 80% OEE, so then somewhere something is wrong actually, either scientists or we have not integrated. So if we are having above 80% OEE, so we are in a green signal, company is doing well, we are doing well, and we are in a good process. So this is how you need to review, and you have to take a call and assume if some of your facility is underutilized, and you try to go for next product which can be filled in same facility. So again, you will end up in manufacturing few products in a same facility. So that is called a multi-product facility. Say for example, if we have 100 products, so we cannot have 100 dedicated facilities, which is going to be very expensive. So in this scenario, so you can have the similar products in same facility, but again there are different requirements. I will cover in my next slides how smart you have to what the key factors that has to be considered if you have to go for multiple multi-product facility in same multi products in same facility. Okay, so now I will just go into formulation part little detail with some case studies, and one is current vaccines. So only adjuvant approved by US FDA and aluminum, the US FDA and WHO is aluminum adjuvants. There are 2 types of aluminum adjuvants, aluminum hydroxide and aluminum phosphate. You take any vaccines which are available in market. So it's going to be with either aluminum hydroxide or aluminum phosphate. So aluminum hydroxide is nothing but aluminum oxyhydroxide, and aluminum phosphate is aluminum oxyphosphate. Okay, so I will just give examples how different how different this aluminum hydroxide and aluminum phosphate in my next slide. So now, as I said earlier, the NDDS based adjuvants are being developed. So I will give 2 case studies. One is linked with the particulate delivery system that can be either microparticles or nanoparticles or dendrimers, or vesicular systems which can be either liposome or neosomes. Then there are other so adjuvants are being explored. As I said, the transdermal patches with the TLR agonist MF59 ISCOM QS21 MPL adjuvants. Okay, so as I said, now there are multiple adjuvants are being explored to increase the immunogenicity, to cover the immunity, and the existing adjuvants, existing formulations are mainly for humoral immunity. So either you have 2 options, either you can buy this adjuvant ready-made from XYZ companies. So only limited companies, and all over world only 2 companies are manufacturing. If you have huge capacity and if they cannot afford to supply enough adjuvants, you have another option to form preformed gel or in situ precipitation, and it is not ideal process. You have to manufacture similar adjuvants in all process. So how smart, how your process is robust is very important. So now I will just differentiate between aluminum phosphate and aluminum hydroxide. So aluminum phosphate, as I said, it's amorphous aluminum oxyphosphate, which is having point of 0 charge. It is nothing but isoelectric point between 5 to 7. So less than this PZC value, it's going to be negative charge, and above the PZC value, it's going to be positive charge. And if any adjuvants which are having opposite charge in this range near like 7 pH, so if you are choosing the IEF in alkaline, so here it gives 100% adsorption. The percent adsorption is nothing but the antigen is adsorbed onto aluminum adjuvant. So this will be adsorbed by electrostatic attraction. Okay, so sometimes what happens, you will be, when you go for combo vaccines, 6 antigens, one will have isoelectric point in acidic range, the another will have in a basic side. So how you are going to keep different characters, means one guy from Japan, the another guy from Europe, the another guy from US will have same culture, so it's not going to be same. So how to keep them in one environment, and how to how to make them stable so that you will have long shelf life is the role of formulation scientists. So you have to perform a lot of pre-formulation studies to ensure that the different physicochemical properties of different adjuvants are intact in X condition. So you have to identify and you have to ensure these formulations so that your formulation, your vaccine is stable for long shelf life. And aluminum hydroxide is nothing but aluminum oxyhydroxide, and the PZC value of this aluminum hydroxide is 11. So at physiological pH, it's going to be positive charge. The positive charge, positively charged at physiological pH. Okay, then the primary particle, that is the particle size also I have just given. So you see this antigen adjuvant adsorption. Generally, it can be electrostatic attraction or ligand exchange, or how it is getting adsorbed under aluminum phosphate is very important. Okay, so if you see this graph, actually zeta potential and the pH. So the blue color one is the aluminum phosphate. So if the pH is less than 6, so it goes towards positive side, and when it comes to about 5 pH, it moves towards minus side. Okay, it is going to be negatively charged, and vice versa for aluminum hydroxide. So when you develop formulations, so how smart enough to take this aluminum hydroxide or aluminum phosphate is very important. And there are some cases, for example, hepatitis B, if you check, and which is having isoelectric point at 5.3, and if you directly adsorb onto aluminum hydroxide, which is having isoelectric point 11, so you will get 100% adsorption. But if the the antigen adsorption, the binding efficiency, if it is so strong, it will not, the adjuvant will not allow your antigen to get expressed to antigen presenting cells such as dendritic cells, macrophages. So the too tight binding also is not good actually. So you need to have strategies to avoid this tight binding efficiency, so that it get presented when we inject our vaccine. So there are multiple factors which have to be integrated when you develop your formulations. So now I'll just start with some case studies. As I said, I will start with some particulate systems, so that is microparticles. Then in microparticles, we have multiple polymers. So I will just present with two. One is natural polymer, that is chitosan, which can be cross-linked with either TPP, that is tripolyphosphate, or glutaraldehyde saturated toluene as a cross-linking agent. And the another synthetic polymer is PLG, a polylactic co-glycolic acid, or polylactic acid, or polyglycolic acid. So PLGA is nothing but the combo. The glycolic acid is meant for immediate release, and lactic acid is meant for sustained release. So if you want to take a balance, you have option to choose PLGA. Again, PLGA, you have different combination, 50-50, 50% glycolic acid and 50% lactic acid. So depending on your requirement, your antigen profile, so you can choose either PLGA or PLA or PGA or PLGA combinations. Okay, then vesicular systems, I will also give like liposomes and neosomes. So again, vesicular system, you have option to manufacture in positive charge, negative charge, and neutral charge, depending on your requirement. So the systems which I present today, I will just show with two things. One is PLGA based microparticles, and PLGA will have negative charge. So the route which are selected for administering these vaccines are nozzle. So nozzle mucosa will have salicylic acid, then it is going to be negative charge. So if you are giving negatively charged microparticles through nozzle route, so it may be cleared. So you need to have positive charge, positive modified PLGA microparticles which will have positive charge, so that the nozzle clearance rate will be slowed, like you will have longer time in the nozzle cavity, so that it gets presented to Nolte cells, that is nothing but nozzle associated lymphoid tissue. And the another one is liposome in situ gelling systems. The model antigen which I present today is hepatitis B, so it's are in public domain, which is easy to present. So we have a couple of other results which I cannot present since it's a confidential. So I'll just start with this PLGA or this liposomes. All these things are like old systems. It's being explored like in many like last 30 years. Okay, so now what were the challenges, why it was not successful? So one was low entrapment efficiency. Okay, the second one means if you entrap 100 microgram and you get entrapped, like you get 10%, so means 10 microgram will get entrapped. If it is recombinant product, if you get 10% efficiency, so how is it going to be practical? So it's not advisable. Then risk of altering antigen integrity. When you have PLGA, so when you store it, the PLGA get degraded into lactic acid and co-glycolic acid, which induces acidic environment inside PLGA microspheres. So the antigen integrity gets deterred and it will not give actual antigen response, means it will not be presented. And due to this acidic environment, this PLGA, the antigen is not going to be stable. So in this scenario, what it was done is, so the low entrapment efficiency has to be improved, then acidic environment should be neutralized. So once you are successful with this, the proposed system can give both humoral and cell-mediated immunity. Means the existing system with adjuvant gives only humoral immunity, that is nothing but IgG. And if you can present through new adjuvants, which follows both MHC1 and MHC2, so it gives cell-mediated immunity plus IgG humoral response. So let us compare this with conventional vaccines, how it has behaved. So you all pharma students, you would have studied with emulsification process. So PLGA with double emulsification methods with polyvinyl alcohol, we prepare secondary emulsion, that is nothing but water in oil in mortar. So the PLGA, I'm not going to the detail in about this, how we formulated and these things. So in the current systems, we have explored more than 150 protein stabilizers, and we have chosen one stabilizers which give good protection. This excipient is approved by all regulatory bodies. And you do have antacid, that is magnesium hydroxide we have chosen, and which avoids the acidic environment inside the PLGA microspheres. So we integrated protein stabilizer and antacid, and we prepared this modified PLGA, which gives positive charge. Another one is in situ gelling system, where phosphatidylcholine and cholesterol are dissolved in methanol and chloroform, and the thin film was again dehydrated with antigen and polyacrylic acid to form in situ gelling system. So I'll just give with examples. When you make up for formulations, there are different methods to identify whether antigen integrity remains same. I'll just start with SDS page. This is SDS page with the silver staining. And you have 4 land. The land 1 is plain hepatitis B, that is controlled bulk antigen, that is nothing but API, drug substance. And the land 2 is modified hepatitis B with triggerlose as a stabilizer. You see the land 1, the intensity and the path, the molecular weight of the formulated like hepatitis B in microspheres are comparable with the land 1, that is controlled. And land 3 is in situ gelling system, and it is also comparable. And you see the land 4 where we have not used the stabilizers. So it's the same process. If you don't use stabilizer, how protein is getting deterred. So it got degraded. So the stabilizers play a huge role in stabilizing protecting the antigen integrity. So in addition to this SDS, there are other methods also integrated to ensure the physicochemical properties like isoelectric focusing, Western blot, TEM, relative potency, and some more. And I just compare this to liposome in situ gelling system and modified PLGA with different formulations. So if you see the zeta potential, as I said, the PLGA, plain PLGA will have negative charge, so that is nothing but minus 0.8.7 with a size of 5 micron, and the percentage entrapment efficiency was 75. If you don't integrate triggerlose, you will get around 10 to 15%. So this is the difference. And modified PLGA, the zeta potential is positive charge, that is 27 millivolts, and the size is 6 micron, and the percentage entrapment efficiency is 75, and in situ gelling system is around 50% entrapment efficiency, and it is negative charge. And you should keep in mind, whenever you give with vaccines, So it is always the preferred optimum size, particle size for better antigen presentation is 1 to 10 micron size. If you don't maintain this size, even if your product is good, if your antigen is good, it may not be presented well to the antigen presenting cells such as dendrite, pair patches or dendritic cells, or it will not be presented to antigen presenting cells. So the keeping the homogeneity, the optimum size between 10 and 1 to 10 micron is very important and key in developing formulations, mainly for vaccines. And as I said, the nozzle clearance, if you compare this lactose, which is green color at lower end, so we used it's a negative control, how fast it is getting cleared from the nozzle cavity. The second one is blue. It is nothing but negative charge. When you go for nozzle activity, it's a percentage radioactivity remaining in nozzle cavity. Okay, so then now if you see this blue color, it got cleared, and compared to negative control, it is getting cleared in the nozzle cavity at like faster rate. And if you go for in-situ gelling system, that is red color, it gets slowly. And if you go for the last top one, is modified PLGA with positive charge, so it has longer time. It remains in nozzle cavity for long time. So this shows that the top two are considered for the further studies, which we expect this should have better antigen presentation to it remains in nasal cavity for longer time. I'll just give you examples. When the PLG is negative charge, when you take this vaccines, what happens? Since it's a negative charge, mucosal, it will have negative charge. It will get cleared. Your product is intact, vaccine is intact, your antigen was stable, but still it gets cleared and it will not lead to antibody protection. And when you go for modified PLGA, your nasal mucosa will have negative charge and your system is having positive charge. So through electrostatic attraction, it gets bound to this one and it leads to, followed by antigen release and presentation to antigen presenting cells. And finally, antibody gets spread, and it induces antibody. So this is the difference. Though you had formulation, only surface charge had impact on this immunity profile. Then the another one, in-situ gelling system, when you take these vaccines, then it through mucosa, it forms a film, thin film. Then it gets presented to antigen presenting cells. Then it induces the antibody response. It's just pictorial form for easy understanding how it gets presented in nasal mucosa. And if you see the IgG titer, as I said, so we have the existing conventional vaccines, which is aluminum based vaccines, which gives good IgG titer. If you see this curve, the longest one, the purple color, gives maximum, that is LM adsorbed hepatitis B with its conventional one. It has given very good IgG titer. But if you see the PLGA based on the LIGS, the lowest one is the PLGA based microsphere. Since it was negative charge, it got cleared from the nasal cavity, which has not given better significant IgG as compared to control, that is aluminum adsorbed hepatitis B. And if you see the other select systems, that is modified PLGA and in-situ gelling systems, so though IgG titer is not superior to the existing one, it can be comparable or equal. I will not say equal, it's comparable when compared to the conventional vaccines, that is aluminum phosphate. So means the developed select formulations could induce comparable IgG titer as compared with the aluminum adjuvants. If you come back to cell-mediated markers like IL-2 and IF and gamma, you see the aluminum adsorbed hepatitis B, that is the conventional existing marketed vaccines, gives low IL-2 titer as Sorry, I got a call. 1 minute. Fine. So IL-2, if you see this aluminum hydroxide has given very low IL-2 titer, and PLGs being negative charge, it got cleared from nasal cavity, it didn't give IL-2 response also. So then if you see this, the select systems, so IL-2 in spleen was significant as compared to other, marketed preparations. So it shows that even in, if you see the second one, IF and gamma in per microgram, nanogram in per spleen, if you see the select system has given significant IL-2 and IF and gamma titer as compared to the conventional vaccines or existing vaccines. And in addition, this, the select formulations, that is modified PLGA and the in-situ gelling systems, also induced secretory IgA. in all mucosal surface. This was given by nasal vaccines. So we could see the secretory IgA in all mucosal surface, that is vaginal secretion, nasal secretion, salivary and serum. So it shows that the immunization in one mucosal surface gives mucosal immunity in all mucosal surface, which is very good actually. So since the vaccine, most of the infections end up through mucosal surface, so inducing secretory IgA in all mucosal surface is it's ideal expectations. So we could see significant increased titer with the new adjuvants. So our conclusion is the select systems which follows both MSC1 and MSC2, so which could induce both humoral immunity and also cell-mediated immunity with secretory IgA. So in short, it is highly muco-adhesive vaccines. It can be called as mucosal vaccines. So we could see the antigen loading up to 75% and antigen integrity maintained upon encapsulation and release. It has given good stability up to 2 years. And as I said, it could induce both humoral and cell-mediated immunity. So now I will just give some examples of cost-effective strategies that we should consider when you develop vaccines. So as I said, actually, I will just give examples. So this is one of the products meant for oral vaccines, as oral vaccines. So some company, when they started, so it's being oral vaccines, what happened, this vaccines, when you take oral, the drug substance, when it gets exposed to acidic environment in stomach, the virus titer drops. So it will not act as vaccines. So you have to take antacid just prior to your vaccines, oral vaccines. So some company, they started developing antacid and also vaccines. So they had two presentation, antacid in one vial, the another vial vaccines. So when they supply, so they have to fill antacid in one filling line, and they need to have one facility for antacid. They need to have another facility to manufacture vaccines. And when you pack, you need to have 2 vial, 2 stopper, and 2 administration, antacids and vaccines. So this is, some company came up with this strategy. There are some company, they came up with all-in-one vaccines. That is, it has both antacid and vaccine in one vial. So it is not easy. When you mix with antacid, it will not be stable. So you have to take a balance. What type of antacid should be taken? What is the acid neutralizing capacity? What is the acidic environment in infant stomach? And how much acid gets secreted? So you have to integrate and you have to prove it that virus titer remains same and also it is having acid neutralizing capacity. So some company worked with all-in-one vaccines and they came up with liquid vaccine. Here you need one team to manufacture one vial, one rubber stopper, one presentation, and when you have in two pack, your space will increase, packaging, cold chain management will be difficult. So here in all-in-one vaccine, you have in single vial. Then another company who developed vaccines, antacid in liquid form, then the vaccine was lyophilized. Since the vaccine, the virus was not stable, so they prefer to go ahead with the lyophilized one. Then some company, the liquid vaccine, they still try to fill in BFS system, that is blow fill seal system. So who will be the successor? Who can be, who can compete well with other companies? So guys who had with lyophilized, COGM will be more as compared to the other two guys. Then the antacid and vaccines will, the COGM of the first one will be higher as compared to all-in-one. So how smart you choose is very important. So if all three guys come in market, you see who can manufacture more product, whose product will be very expensive. So when you develop, once you do clinical trial, again you cannot revisit and do another trial which is going to drain your money. So how smart you choose your strategies, how you choose and how you manufacture it, how you define your process, how you ensure the multiple clinical trial is very important. So this is very important. So I'm not going in detail, some things are confidential. So we have to avoid process change and multiple clinical trials, and you have to choose your facility and product considering market demands and OEE, COGM and quality requirements. And we should have a balance. So if you concentrate only market demand and if you lose another parameters, then it's going to be setback. So you have to take a balance and you have to consider multi operational requirements. And the process knowledge is obviously very important. And in commercial, it's a biological process. The way it behaves in summer, it is not going to be same in winter. and sometimes you will have, you will not know what has happened. So in this scenario, if you don't have right people, and if you don't identify, if you keep your facility ideal for 1 month, say for example now COVID, if you have some problem, if you don't fix on same day, if you keep ideal for 1 month, how much it's going to be our setback to everything. So how smart to fix your problem and how you have robust process is very important for cost effective strategies, and which is very important if you have to sustain in a market with the 10 companies. So you have to consider many points so that you will be the best person, you can be right successful first time. Okay, so the level of the sterility also very important. If you are going for open vial, the sterility level risk is high, and open ampoule, it comes down with closed vials. As I said, this isolator, now in serum we have completely closed vials based operations, and we have like completely closed and the automatic systems. So we are into almost negligible risk in our vaccines with respect to safety or the sterility practice. So this is how we operate it. You'd see how it evolved from 30 years back, and currently where are we. So for the now I have 5 more minutes to complete. So the pharmaceutical aspects and manufacturing challenges, as I said, you have to develop simple process, and you have to take a balance, liquid and lyophilized and excipient and stability, and R&D scale, pilot scale, commercial scale, COGM, and product equivalence is very important. As I said, if you don't have equivalency, then regulatory bodies will not accept. So you have to defend your product during your audit. So unless you have strong CMC data, it's not going to be your, you cannot have cakewalk. So you need to have strong team and also the data to support, to defend your product facility and your team. And process validation, obviously, it's very important. So generally we do with 3 or 5 batches process validation, and we saw that the batches which are prepared continuously, we are complete, like robustness, we consider 50 batches. And process validation also is very important, so that our product are reproducible and it has consistent process. And mixing and homogeneity is very important. Say for example, when you have 50 liter vessel, and how you ensure homogeneity strategy is different. When you have a 3000 liter, your minimum stirring volume is going to be 250 liter. So the last 250 liter vaccine cannot be discarded. So what strategy, how you are going to keep them homogeneous, because it's a suspension, it tends to settle. So how smart enough, and you should not waste even 1 ml of vaccines. That is the requirement. So how smart you develop your facility, you develop your equipments, and how you ensure the homogeneity and safety and efficacy in your product is very important. And as I said, if you have multiple products, the cleaning validation is very important. If you manufacture A product, before you manufacture B product, you have to ensure that A product is not existing in your vessels. So there is something called cleaning validation. This is another topic which we'll need to speak for a few days to talk on cleaning validation. So there are regulatory expectations, you have to fulfill this. And in filling, you have options to have them either in single dose vial or multi-dose vial. The multi-dose vial can be 5 dose, 10 dose, or sometimes 20 dose. So ideally, multi-dose vial will have with some preservative, or you can also have PFS, pre-filled syringe, and mostly we prefer for private market. And in short, the importance of formulation and manufacturing strategy in vaccine industry is critical. It's a biological process. It's not going to be cakewalk. So you need to have a right team, right facility, right management, like whatever we need, and management also should support. So we have the best facility in the world, and we are confident we have right team, and we will be able to deliver right first time. And novel delivery systems are very important, and way forward it is going to replace the existing adjuvants. So vaccine industry is going to evolve further, and we will have new technologies, new adjuvants, so that it gives broad protection. And cost effective strategies are very important. There are 50 companies in queue. So how smart you define it, how you smart commercial batches, how you manufacture it, how you occupy your facility, so which helps to be competitive, to be competent with your competitors, or to be competent enough to sustain in market, survive in market. It is very important to integrate cost-effective strategies during scale-up and commercial manufacturing. And commercial also, like if you have some issues, you need to have right team to go for troubleshooting, and you have to ensure robust process. The product, if you manufacture 100 batches, all 100 batches should be comparable, and the physicochemical parameters of all batches have to be comparable. And continuous quality improvement and regulatory integration is very important. So the current quality expectations and regulatory expectation may not be same in 2022 or 23. So when something is getting integrated, you have to also upgrade your facility, your knowledge and your manpower to the quality requirements and the current regulatory requirements. So thank you very much for your time. I think I have taken 10 minutes more. If you have any specific questions, I'm free for the discussion.
Thank you very much, sir. And it's time for question answer. It's a good opportunity. You have got Jagan sir with us to answer our queries. Yes. Jagan sir, may I request you to open your camera? We are not able to see you actually.
Sorry?
We are not able to see you. Can you open your camera?
Camera, okay. Yeah. You can see.
Sir, thank you. Yes. So, any questions from the participants? But sir, I have got one question.
Sure.
That how, particularly in the light of this corona vaccine, I want to ask that how we can make our corona vaccine suitable for more and more strain. Because last day I was hearing in news on ZTV that US has approved one vaccine that it is effective against 600 variants of the coronavirus. So what has to be taken care that our vaccine should cover more and more strains of the virus?
Vivek, there are multiple factors to be integrated during development. So, for example, even in serum vaccines, if you consider this virus is getting mutated, so you have different strains, and the epitopes, the small change in one amino acid should not change the complete, these things. So I think now you have to, it has to be considered from development. You have, for example, to be specific in our COVID vaccines. So if you take our vaccines, maybe 5-6 months back, so it also had, it good protective efficacy against multiple variants. So this all depending, for example, we have integrated spike protein. Okay, so the one small mutation will not change your vaccine completely. Okay, so it gives protection even if you take the guys who get infected after any vaccines. So what happens, they are not moving to any though they get positive, they're either asymptomatic or mild symptoms, very mild symptoms. So it gives protection. It is not like completely zero. So this is very short time to link with the COVID vaccine. In ideal path, if you have to develop and go for complete, the CMC and clinical science, it generally takes 8 years. So in 2 years, and being specific with the antigen, how to integrate will be little challenging. So maybe we have to wait and have more data to see the exact, to express our, the exact reason how it gives and how to integrate. So everyone they consider, say for example, some they consider mRNA based, some we have considered spike protein with the chimpanzee, and the other companies are inactivated. So we have to see this. I think some are confidential. We cannot also speak in open forum.
And sir, in fact, launching vaccine, corona vaccine in just 9 months is a big, I should say, success for the Indian pharma industry. So sir, how you are able to compete with the international pharma vaccine industry actually? So they have got very good R&D, but from India we have got 2 companies. And within a record time, they are able to launch the vaccines. And not only they are able to launch the vaccine, but the efficacy was very good. That was even better than the internationally available vaccines, which are there in the market. So how could you do this, sir? It's a big achievement, sir.
It's a teamwork, Vivek, actually. So this all, again, I should not speak more about COVID vaccines in the public forum. So this all, we have smart team, and we have best management, and we have best facility. So we are successful. And the filling machine, for example, what we have in India, what they have in abroad are same companies. So I don't see the Indians, we are flexible. We work even if we come, sometimes we work up to 10 hours also. So we are fine and we are dedicated to science. And in other companies, it's not going to be, other countries, mainly in some European countries, and they have union. I just give examples. In one of the European country, their annual number of working days in their country is 150 days per year. So in India, you just compare, where are we? So what they do in 1 year, we can do it in 3 months.
Great.
Hello, sir.
Yes,
Thank you very much, sir. I do not have any question, but I must tell you that thank you so much. The presentation was excellent. You have covered all the aspects of the topic, and we are very much thankful to you. In spite of your busy schedule, you have taken your precious time to us. Thank you so much, sir. It was really very nice session.
no problem, madam. Thank you so much. Thank you for giving me an opportunity to present it. It's always pleasure to talk, madam, in academics.
Thank you, sir. Sir, over to you, Vivek, sir.
And sir, as a token of appreciation and gratitude from the Parul University, we present this certificate to you. Kindly accept this certificate, sir.
Thank you.
We had been fortunate to listen from you, but since it's a Corona period, so everything we have to do virtually. But thank you very much, sir, that you spare time and come over here.
No problem.
And giving us a deep insight about the vaccine formulation and the smart considerations which has to be taken care of while formulating the vaccines. Thank you very much, sir.
Thank you, Vivek. Always pleasure. Thank you so much.
And in future also, sir, we would like to hear from you on these interesting topics.
Sure. No problem.
Thank you very much.
Yep.
Thank you.
Bye.
So this marks the end of our webinar, and I will be sharing feedback link to you in the chat box. So you are all requested to give your feedback on the webinar so that we can make our future webinars more and more interesting and more engaging. Thank you very much, all of you.